All right. Hello, good morning, good evening, everybody. I am Vaidya Asavari Manvikar. I would like to welcome you all to GCAR's Global Webinar Research Series. GCAR, Global Council of Ayurveda Research, is an non-profit organization based out of USA. Our mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence based health science globally. You can learn more about us at www.ayurvedaresearchusa.org. Please do consider joining us as a member, which will bring you access to all our resources. No nonprofit can operate and succeed without volunteers. So consider joining us as a volunteer, and we can place you in a variety of projects based on your interest and skills. Once again, our website is www.ayurvedaresearchusa.org. Our eminent speaker, Dr. Mitali Mukerji, is here for our second session. Thank you so much for joining us regarding this session. So let me hand it over to Dr. Mitali Mukerji. She will be guiding us in this second session regarding the future of personalized Ayurveda, the Ayurveda genomics.
Thank you. And good evening, good morning, wherever people are hearing from. Shall I share the screen?
Yes, please.
Yes. Okay. Okay. So, hello everybody. This is the second lecture in the series on Ayurgenomics, and I'm going to do a brief recap of what we discussed last month rather, why the need for Ayurgenomics and how we went about initiating this activity. And it emanated from this gap in the modern sciences, where the first aspect which I described was the health longevity gap. People are living healthy, but meaning people are not dying early, but they are not in the last 10 years of their life. They all are suffering with one or the other complex disorders. So the lifespan has increased, but the health span has reduced. And the Human Genome Project was an initiative to understand the various aspects of health and precision medicine. So the idea was how you can understand a genome from its first principle so that you have a more personalized, predictive, preventive, as well as participatory approach to medicine, which is what is now being called as P4 medicine, and how these resonates with the Ayurveda principles, which have been in there for the last 2,000 to 3,000 years, and how many of these aspects of network medicine, systems medicine, personalization, participatory aspects, these are all well described in Ayurveda, and how and the question was whether we could actually integrate this with the current perspective of P4 medicine, so that we might have a much better integrative approach to medicine. And in order to do that, we first need to bridge the ontologies. We need to understand something which is written in a totally different language and has a totally different epistemology compared to modern medicine, how one can go about integrating it. And I think left it at our first study, where we first went around showing the molecular and the transcriptomic correlates of Prakriti, of predominant Prakriti, which have the enrichment of either of these doshas, and how they correlate with Ayurveda descriptions. And so that's what I called as Ayurgenomics. So it's not one way integration, but it has to be iterative, and that's what we were discussing. So this is the framework which I will take you through the classes. So the first was how to develop a working hypothesis from the principles, the common organizing principles of Tridosha and Trisutra. And if you have developed a working hypothesis that if doshas are described as kinetic, potential, and metabolic component, then how do we query them? Could we use the language of genomics to query them? And if we get them, could we also recapitulate them in cell models based studies? And I had shown you one such example of cell proliferation, how it differentiates Vat and how it governs uh, uh, differences in DNA damage response axis at the cellular level, and is correlated to phenotypes in diseases which also correlate with what Vat Prakriti is described as. So now, given that there is this correlation, does this correlation have a genetic basis is what I would like to take you through this time. And just before going there, I'll just take a little detour that how these genetic variations pan out in the population. So 100,000 years back, people moved out of Africa from different parts to different parts of the world. They colonized different regions, and there were many selective pressures like climate, food source, pathogens, altitude, aridity, latitudes, and all these would have an impact on our meaning would impact, and there would be selection for who gets to thrive or survive in these areas. And besides that, there also have been lots of evolution of cultural practices. The first was our ability to control fire. So that gave us very different digestive capabilities. Earlier, people were hunter gatherers, so the food would have been uncooked. But then, when we learned to control fire, the food was more digestible. So that led to differences with respect to our gut. Then there were other kinds of things which are like moving to different kinds of sustenance. For example, farming, industrial revolution, and all these would have impacted us in one way or the other. And these can be very evident. One of the very prominent example is how the prevalence of dairy farming is correlated to lactose tolerance across the world. So the populations which are hunter-gatherers, they still remain largely lactose intolerant, whereas the different parts of the world where cattle was bred, dairy farming was there, lactose tolerance set in. And similarly is the case of domestication of wheat and prevalence of celiac disease, because as we domesticated the wheat, they became hexaploid, and that led to more prevalence of celiac disease in different populations. So these are observations on disease prevalence vis-a-vis different populations. But how does genomics come in? What does genomics tell? So, as I had described, that after the human genome got sequenced, one of the most surprising observation was that we vary quite a lot. Even with two related people, we would differ at a million places, and there are millions of different kinds of SNPs. These could be single nucleotide, these could be insertion, deletion, a block which could get added, inserted, and then there's something like copy number variations, that is chunk of genomes or chunk of genes, they can get duplicated. And some of the interesting examples are, for example, there's this salivary amylase gene, which has expanded in some of the populations which have moved to starchy diet. That is because they could digest more of that food which they were dependent on. But that same variation is also linked to obesity in populations. The other thing which the genome differences reveal is the history of migration. For example, there was a study, a very prominent study, it was called the Human Genome Diversity Panel, where they looked at populations in different parts of the world and tried to correlate how they are closer to each other or proximal to each other by these variations. And they found that as the populations move out from Africa to Middle East to Europe, Central South Asia, East Asia, the extent of relatedness keeps on getting reduced. And within a continental population, many populations are related to each other. But in some populations, there are also signatures of admixture. That is, there are some kind of, and it's also based on the proximity of these geographical proximity of these populations. So if you see in this panel below, there are all these vertical lines. These are all individuals from various populations, and one color represents how closely related they are. And if there are two colors, then you can make out that it is also admixed with a population which is like Middle East. Many populations are more closely related to European population. Central South Asia is very distinct from Europe, and East Asia is quite distinct, with a few population admixed with Central South Asia and East Asia, and then Oceania and America. So how people have moved, the genomes have changed in that manner. And this is relevant because this ancestry also tells you that if there are mutations in the populations, they can also get carried by these waves of migration. And it's very important, that's why, to contextualize any variation in the background of the in context of the genetic background. So similar thing we have observed in the Indian genomes. I talked about Indian Genome Variation Project, where Indian populations are extremely diverse. They come from different linguistic lineages. And we had provided the first comprehensive map of the Indian populations, where we showed that the Indian populations map into 4 broad clusters. And why is that important is something which I'm showing in the right side of the slide, that you see malaria prevalence map on the left, and the right side has a spatial map of genetic polymorphism. And you can see the population which are endemic for malaria. There the frequency of a particular gene differs very drastically as compared to the others. And then we went around asking a question that if this is different, are these variations actually protecting these populations because they are residing in malaria endemic region? So we tested them in malaria susceptible and malaria protected individuals, and we find that these variations are linked to protection from malaria. So such kind of background data allows you to contextualize variations and ascertain whether some of the variations which are there in certain populations actually give an advantage for them to survive or thrive in the population. Similarly, there's a genetic variation. It's called CCR5 delta 32. This is a variation which is linked to susceptibility to HIV. So there is a mutation in this gene which does not allow HIV to enter. And this protective allele is only present in the population which is most closest to the western frontier, which is the route for most of the migration from the western front. And they are most protected from this, but rest of the country seems to be at risk, at least with respect to this variation. So, and then genomic variation, as I had also described, is also being integrated in all aspects of how during fetal stages, newborn, undiagnosed diseases, how if there are variations which allow you to predict who are the people who would be more prone to diseases in cases of cancer, detecting predisposition, pharmacogenomics, also predicting health span, and then from the molecular autopsy, the cause of death. The other aspect, which also is gaining very prominence, is how variations are linked to our dietary behavior. So this was an interesting paper on you have a burger with different kinds of things in them, the meat, the cheese, and then tomato, onion, and each of them have some chemicals which are perceived by these taste receptors, olfactory receptors, and which govern how what you would like to have, meaning what you perceive at lower threshold and what you perceive at higher threshold. And that's the reason maybe that people, given the same kind of food, they would perceive it differently and they would want it differently. So this is what would also then govern that once you have you there meaning this would govern inter-individual variability in dietary behavior, which would also then be linked to variability in predisposition. So if you want to understand human individuality for precision medicine, there are three aspects. One is the heritable component, the other is the environmental component, the response component, and the third is the interventionable component, which is modifiable by diet, therapy, and lifestyle. So I say understanding who I am is very important for precision medicine. I is innate, A is acquired, and M is the modifiable component. So now, from this, how do you get to the Prakriti and genetic link? That we see differences amongst Prakriti type, but is that correlated with genetic variations? How do we go about finding that? And so the thing we observed was predominant Vata, Pitta, and Kapha exhibit differences at the transcriptome and biochemical level. So could these differences be governed by variations at the genetic level? And uh, if these differ, then do the dosha associated genetic variation confer differences in phenotypic differences or differences in disease susceptibility or responsiveness? So that's the question we started asking. So in that paper, which I described in the last lecture, we observed from a North Indian cohort when we did the transcriptome analysis in predominant Prakriti, there were a large number of genes which differed between the males and females between the Prakriti group. So we decided to ask whether if we take the variations in these genes, do they differ between the Prakriti type, and would those differences have some meaning? So in order to do that, the first thing I had mentioned was that these differences should not be there just because the background, they come from different backgrounds. So we had to ascertain that all these Vat, Pitta, and Kapha individual come from a homogeneous genetic background, and the differences we see are because of the inter-individual variability within the background and not because of the differences conferred from the other populations. So that was the first thing we did. So what we did was we integrated our data with data from 24 populations across the country, and showed that all our samples were derived from this blue group, which is the Indo-European people from northern part of the country, so that they are not confounded by the other populations. So in Ayurveda, it will be called as jati prasakta, or or meaning that's the aspect we wanted to kind of negate here. So now we work with very extreme phenotypes, which comprise only 10% of the population and who are more prone to different kinds of diseases. And we looked at genetic differences in them. So what we did was we handpicked around 30 genes which differed at the expression level, and we did the genotyping. That is, you amplify that region which has that single nucleotide polymorphism. These are typically represented by a number which starts with RS. So let us say this is the gene. It's called AKT3. It has an SNP which has a variation C or T. And this gene is highly expressed in Kapha. Now, when we look at the frequency of this variation, it differs between Pitta and Kapha with respect to the C allele. So such that the allele frequency of C in one, that is P, is 28%, and it is 58% in the other group. That means there is a significant difference in the frequency of the C allele between Pitta and Kapha in AKT3 gene. So like that, when we took 30 genes, we have to do some kind of statistical correction. And then we found that there were 5 genes which came out very different between the Prakriti groups. some between Pitta and Kapha, some between Vata and Pitta, like that. So there's this gene linked to DNA damage and repair, and it's linked to breast cancer and many other cancers. This is a gene which is linked to a disease called Marfan syndrome. This is a gene which is linked to a process called apoptosis. Then this AKT3 gene is also linked to melanoma. And this was something which was interesting, EGLN1. At that time, not many people knew what this gene is doing. So we decided to, and then the most important thing is that if you look at the background population, that is a population which has not been stratified by Vat, Pitta, and Kapha, then it would have a heterogeneous mixture of Vat, Pitta, and Kapha, as a result of which the same allele would get have an average frequency. So, for example, for this gene FAS, if the allele T is lower in Kapha and higher in Pitta and lower in Vat, if you pool all of them together, they have an average frequency, which is like the background frequency. So what does this tell is that if you stratify a population, just like we showed with respect to biochemistry, at the genetic level also, if you kind of classify them by phenotype and look at genetic differences with respect to certain genes, they show differences in allele frequency, which gets homogenized in the pool population. Now, how is that relevant is what I will tell you. I'll tell you a story with respect to this gene called EGLN1. This was a very interesting gene to follow. This the frequency of one of the allele is low in Kapha and high in Pitta, and also reasonably high in Vata. And if you look at the VPK, if you pool all of them, it has an average frequency which is like the background Indo-European population from where they were derived. Now, what does this gene do? This is an oxygen sensor gene. This gene, in the presence of oxygen, becomes active, and what it does is it switches off a set of genes which are needed to cope up with low oxygen condition. So we typically are not living in low oxygen conditions, so we don't need to switch off a large, we need to switch off a large number of genes which we only need when we go to a low oxygen condition. So this gene works as a switch in the presence of oxygen. So if oxygen is not there, this gene is not active, as a result of which, by default, all the genes related to coping up with hypoxia get switched on. Now, this differed between Kapha and Pitta very significantly. So the question was that if an oxygen sensor gene differs between two Prakriti type, will that also mean that they would be differently responsive to hypoxic condition? Now, something very interesting we found, because we were also simultaneously doing Indian genomes, and this variation was also included in the panel of Indian genome. So when we projected them in the map of India, we ended up finding that whatever was high in Pitta seemed to be fixed in high altitude populations of the country, and they were irrespective of whether they were from Indo-European or Tibetan background. They were more close to each other, and Tibeto-Burman people who are from the low altitude were significantly different with respect to this variation. So also the Indo-European. So these were Kashmiri people and Leh Ladakhi people. And and then we did an analysis and we found that this variation was linked to the altitude. You see a population around the Gujarat region. So this population has actually come from Ethiopia. We had taken this as a control for an African background, and the population comes from a region which is also a relatively high altitude population. So this is a significant difference between the high altitude and low altitude of the same ethnic population. So if they differ with respect to the altitude, but otherwise, even if they are similar at the genetic level, then they are significantly different. So we asked a question that if this is the case, so in our expression data also we had found that in Kapha there is a high expression of EGLN1, which would say that if you have a high expression of EGLN1, you will lower the expression of genes which are related to adaptation to hypoxia. So the question was that if Pitta-related genotype is more in high altitude people, then would the genotype which are associated with Kapha be more linked to people who go to high altitude and sometimes develop this disease called high altitude pulmonary edema? So this was the question we went about asking, and we were lucky to have a cohort of sojourners who go to high altitude, and some of them developed HAPE. So one of our collaborators was working in that area. So what we did was we collected those samples and we looked at their genetic makeup, and we found that the HAPE population had higher frequency of the allele which was associated with cough, whereas the natives and the pith were nearly similar to each other, and Vat was like a background population. So this actually and the allele which was linked to the higher expression, we could anchor the genotype to the expression levels of the cough also. So this kind of suggested to us that even if you don't take a person to high altitude, maybe if you did the prakriti of a person along with maybe the genotyping of a person, you might be able to predict beforehand who are the people who are likely to do well at a low oxygen condition. So then we also had an opportunity because when genome sequencing are going on, there are also people who are looking at a large number of people with respect to many genomic variations. And luckily for us, the variation which we had observed was also there in one of the panel of data, which had information from different parts of the world, right from Africa to America, the ones which I had shown as the migration pattern which people had traced out using that array. So I looked up the population, and they had given latitude and longitude of these populations, and using Google Earth we could find the altitude, and we found that in some of the high altitude populations in East Asia, again there was a correlation of the same allele with high altitude. So this was like an orthogonal validation in another part of the world, that the variation which we had found to be associated with high altitude adaptation was also the same variation which was seen in other parts of the world. Now, if we go to ancient descriptions of high altitude, that is the Ayurveda descriptions of high altitude, there's something which resonates with whatever we observe. You have very high mountains which are kind of described as Bahu Mahaparvat or Shitidhar, where Kaf, Vat, Rog are more prevalent. So it is anticipated that Pitt might be more protected in these kinds of conditions, right? So this was the paper we published in PNAS. For the first time, we could show that if you use the constitution type of Ayurveda, you could discover such kind of genes which would be very relevant in predicting responsiveness, in this case, high altitude adaptation. Uh, when we were doing this work simultaneously in that timelines itself, many other high altitude populations of the world using different methods arrived at the same conclusion, and it's in the Andes mountains, it's in the Tajikistan, Tibetan, Africa, and India. All across the world, the same gene has been implicated in high altitude adaptation. And in Quechua in Peru, we in high altitude, this gene, this same variation has also been linked to aerobic capacity. So there's a physiological link to this gene. And now hypoxia is not only relevant in high altitude, but it is also relevant in a large number of conditions. And people have shown that if you have EGLN1, it could actually be a very important therapeutic target where hypoxia is a cause or consequence. It's been shown in cartilage repair, wound healing, arteriogenic phenotypes, brain tumor, renal anemia, cardiovascular disorder, recovery from stroke, treatment of inflammatory disease. So that means to say that if people have variability in this gene, then they could also have differences in susceptibility to all these kinds of conditions. And this has been also being shown in many cases of different kinds of cancer, because in cancer, hypoxia becomes much, much more relevant, and it's become a very important drug target. So moving on, we asked that if EGLN1, whether it's one kind of a story or we get many more such kind of genetic differences if we stratify people by Prakriti. So then what we did was we ask the question the other way, that supposing we have a panel of SNPs from a large number of genes, and then we take those SNPs and genotype them in Vat, Pitta, Kapha, do we see differences in those variations? So this was our follow-up study, and you can see we find differences in a large number of genes. Leptin receptor, which is linked to satiety, SPTA1, which is linked to red blood cell traits, UCP2, which is linked to mitochondrial function, OLR1 linked to lipid metabolism and olfactory receptor. And then there was a difference with respect to one of the gene, which was very, very interesting. Again, this is called as a von Willebrand factor. Now, von Willebrand factor differed between Pitta and Kapha. And what we saw, a very interesting thing was we found the same kind of a difference as in high altitude. That is, what we saw with respect to EGLN1 is again what we saw with respect to Pitta. The high altitude natives and the Pitta had the same kind of a genotype, which was very contrasting to the Kapha. So now, is this coincidental, or is there something linked to each other? Because that's a hypoxia axis, and this is a trait. The von Willebrand factor is called as a platelet glycoprotein, and when angiogenesis happens, then VWF levels increase because it's made more to prevent bleeding episodes. But if there is a mutation in this gene, then it is also linked to a bleeding disorder, which is called as a von Willebrand disease. So if you have lower expression of VWF, it is linked to bleeding. And the opposite, that if you have higher levels of VWF, it is actually associated with atherosclerosis. So stroke susceptibility has been linked to one of the alleles. So this is the one which is prevalent in Kapha, has been linked to stroke, and the one which is more prevalent in Pitta is linked to bleeding outcome. So now we asked a question that why we have this allele in Pitta, and is this in some way related to hypoxia? Because if you have angiogenesis, then you will keep on having making von Willebrand factor. Now, supposing you are at high altitude and angiogenesis is a need, and if you keep on making having good angiogenesis, then you keep on making von Willebrand factor, then people will be more prone to stroke at high altitude. So is the bleeding allele, which is associated with Pitta and also high altitude native, does it actually protect high altitude native from not having stroke? Now, how do you go about testing it? So we could not test this in mouse in the humans, but we tested the same thing in a mouse model. So one thing before that, so bleeding as a phenotype associated with Pitta and atherosclerosis as a phenotype associated with Kapha is also described in Ayurveda as an anatmaj vikar. So what we did was we took mouse, and since we know this hypoxia axis works through EGLN1, so supposing if you inhibit EGLN1, that is you lower the expression of EGLN1, HIF1 alpha or hypoxia will be elevated. So it is like you are mimicking the same condition as Pitta in a mouse model. So what we did was we took a small RNA which is against the gene, so that the gene is not expressed, as a result of which PHD2 or EGLN1 is down, HIF1 alpha or the hypoxia related gene will be up. And we measured all the platelet count, platelet activity, all those kinds of parameters we can actually measure. So when we did that, we found that if you make a mouse pro-hypoxic, then the blood becomes so thick that it was also very difficult to draw out the blood, and the platelet count and the platelet activity and even the bleeding time, all of this was measured, and it actually showed that if you make a mouse hypoxic, then there is this meaning if you inhibit EGLN1, that is you make it like Pitta, then hypoxia axis is elevated, as a result of which thrombotic activity is increased. This was also validated by another molecule, single molecule inhibitor, which is called as DHP. So this study actually showed that maybe bleeding phenotype in Pitta is protective if they are more pro-hypoxic, And in cough, because it's not pro-hypoxic, then one can live with higher levels of VWF, but that might also give a risk to thrombosis. So this also suggests that people have different kinds of stroke, hemorrhagic and thrombotic stroke. Maybe if you go about phenotyping these people, would you find more of pit or a cuff phenotype in either of the cases? It is testable. It's not been tested as yet. But now, if you look at these descriptions in Ayurveda with respect to hemostatic axis, these are all given in our supplementary data. How hemostasis is coagulability in cold and cloudy weather condition is also described, and then how effect of lifestyle and dietary practices as well as prolonged exposure to geoclimatic condition and seasonal variation impact hemopoiesis, and how it governs bleeding, atherosclerotic condition. Lots of descriptions are there. So this is where we try to integrate whatever we are observing to whether these also resonate with descriptions of Ayurveda, because as I keep on saying that what we are wanting to do in Ayurgenomics is just to try to see whether we can actually get the descriptions in a molecular language so that we are able to make sense with the contemporary kind of observations which we make. But beyond that, there are also very interesting diet and lifestyle recommendations which are given based on prakriti, habitat, and season. So the idea is that supposing you are of a particular prakriti type and you have to reside in a particular habitat or move to a particular habitat, and if you know your prakriti, would you be able to recommend appropriate diet and lifestyle to keep the person's health in check, so that maybe the pith doesn't get exacerbated or cough doesn't get exacerbated, etc. So, as I had mentioned, EGLN1 has become a very important therapeutic target for a large number of disease. And this also tells that if EGLN1 is a therapeutic target, that means you need to either bring it down or maybe increase it, which is seen in this ischemia, some cases of ischemia where you have to give some hypoxia for recovery from ischemia or something like that. I'm not a clinical person, so but this so EGLN1 is, it's not necessary that you have to always get it down. In some cases, you need to also increase its expression. So one of my colleagues in Delhi, he works in the area of asthma, and he also decided to test this axis in mouse models of asthma. So he had ovalbumin induced asthmatic mouse, which showed all the features of asthma by way of airway hyperresponsiveness, tissue expression, molecular expression, etc. And he found that if he made the asthmatic mouse more hypoxic by modulating EGLN1, then it promotes inflammation and epithelial injury such that the mouse is even more sick than the ovalbumin induced mouse, and it also has gets to be steroid resistant. So this showed that hypoxia could be an important modulator in asthma, and it also would tell you something that if supposing people are of different prakriti, is it possible that there could be more exacerbation of inflammation and epithelial injury if the person is of Pith Prakriti, because the person is more pro-hypoxic, and along with that, if the person is susceptible to asthma, will that lead to much more severe or chronicity in asthma? Uh, then this brings me back to something very interesting, that supposing hypoxia is not good at sea level, and that's why it is there in only a fraction of predominant Pith Prakriti people who would comprise 3% of the population. But if it is fixed in high altitude population, what happens to the Prakriti of high altitude? It wouldn't be that everybody in high altitude will be of Pith Prakriti, right? The other thing is that if you have hypoxia, then many more functions can be compromised. So one of the persons, Sarah Hillman, she's from UK. When she saw our observations, she had a question because she is obstetrician, and there's one very important condition of intrauterine growth retardation, where when you have hypoxia, then the size of the child is smaller. So supposing in high altitude you have this axis switched on all the time, then what would be the birth size of the children? And you cannot keep on having lower birth child. So the birth size of the child. So she decided to test this out. So what was done was, and it was also had been reported earlier that children born at high altitude have uh, a larger in size compared to the ones who are born at sea level. So that means there is something protective in them, so that hypoxia does not reduce their birth size, meaning the size at birth. And so she wanted to know what are those protective, or what are those compensatory mechanisms. So what she did was, with us and with AIMS Delhi, she did a large study where she followed up mothers at high altitude, higher than 3,540 meters, and at low level. And then we took their blood samples, we did their genetic study, and asked a question about what are those protective things in the mother, or which gives rise to this bigger body size of children born at high altitude. And we got some interesting results, and many of them were related to birth weight associated genes and also hematological traits, so that when the fetus is growing in the womb, adequate size and adequate kind of growth is maintained. So this brings me back to something which is also described in Ayurveda as Deshanupatini. It is, we observe that cough kind of genotype are more predisposed to high altitude pulmonary edema, but at the same time, in Ayurveda, it is also described that even though cough vata diseases are more prevalent, the people of high altitude are more of cough prakriti type, which was kind of counterintuitive to me. Why a place which is more provoking that disease, the natives of those places will be of that kind. Now it kind of makes sense that if you have to survive in that altitude, then some things have to be compensated for example the growth and all those things. These are the aspects which are also go to make the cough phenotype, meaning differentiate cough from vat phenotype. So it would be that whenever we are talking about a variation, it has to be contextualized by the genetic background besides the ethnic background, and such kind of results tell us about the Desh Anupatini concept of Ayurveda, where Jangal Desh have more Pitvat, Anup Desh have more Kaf kind of Prakriti. So that baseline Prakriti of the entire population, on which after that you have to assign Prakriti of a person. So you will have to kind of take into account when you are doing a prakriti of a person, that what part of the country or what part of the geography the person is coming from, so that some of those features should not be taken into account when you are assigning vat pit kaf attributes. And the last part is that the hypoxia and hemostatic outcome, differentiating pith and cough, would also have relevance during COVID time, when hypoxia was most relevant. Many of the people were not have had an adverse response to low oxygen condition. Many of them had inflammatory outcome. So it would have been worthwhile maybe to check whether, or one can go retrospectively and check the people who had COVID, the ones who were protected from COVID, ones who had a side effect of the hypoxia, or bleeding or inflammatory outcome. Would there be underpinnings of prakriti in them? So this kind of variability, what I showed you at the genome level, this is being seen at many levels of biological hierarchy. And this is a review article which I wrote in 2023. It actually exhaustively provides information on different kinds of studies being done by different groups where variations have been observed at various levels of biological hierarchy: cellular, genomic, epigenetic, transcriptomic, proteomic, metagenome, even in biochemistry. And such kind of a variability could lead to differences in not only in individuality but also in response, in also systemic outcome. And one could then stratify populations using prakriti. Many kind of these underlying variability could be scaffolded by these bigger prakriti types, and meaning these prakriti types, as a result of which one can have a non-invasive way of endophenotyping people for risk stratification, maybe in settings where one cannot do all these kinds of molecular analysis. But I would like to mention that much of the work over here has been done in predominant prakriti. The idea was to identify from the extreme phenotypes what are the molecular links, and whether those molecular links would also behave similarly in mixed prakriti types, who are 90% of the people, is what is now being tested in our laboratory. So thank you so much, and I look forward to your questions. Thanks. Hello. Did I lose them? Hello. You are muted, Asabri.
Yes, hello.
Yeah.
Yeah, that was a wonderful session, Dr. Mitali. Thank you so much for all the details and the work that you have done. Dr. Smita, do you have any questions?
Thank you so much, Dr. Mitali. It was a really informative session, and I'm feeling like I learned so much, something new, I would say. And then I have questions, but I don't think at the moment, but I would definitely reach out to you.
Okay.
And Dr. Asavari, would you be able to share me the first part as well? So I just want to see that again one more time.
Absolutely. Yep, definitely. I will send it to you, Dr. Smita.
Thank you.
I think there's a YouTube channel where they have posted the first one. Right?
Yeah, so we have GCAR YouTube channel, and we have
Okay.
all the sessions and other webinars also. Everything is recorded, and those are there, that you can definitely watch as well. And
Okay, perfect.
yep. Yeah, so it will be great, like if everybody who is interested into learning more, if they can join our webinars and ask questions and meet, our speakers. And we do have this channel, so please subscribe it and check all the recordings from the previous sessions also. And if you have any questions, please feel free to contact us at Ayurveda Research USA at gmail.com. You can also reach out to me at asavari@mnayurveda.com. And thank you again for joining today. We will be having next session. So we have this 6-week series, like 6-session series for 6 months. So this was our second series and second session. So we will be having our third session next time in April. So this is typically the last Saturday of each month. And we would love to see you back. Thank you so much.
Thank you.
Thank you. Shall I log off?
Yes, thank you so much.